everyone, and welcome to another Hub Cures, number 3 of our series this season, and welcome to a new platform. So, we were using Remo all last year, and I think we were one of the early ones in Boston to use it. Now a lot of people are using it, and we liked it, but it had a lot of technical difficulties. And this company, Goalcast, is a new company. It's 2 Harvard Business School students who dropped out of school last year, when the pandemic hit, and they've created this platform called Goalcast, and we hope this is going to work. I mean, so far it seems to be working well. So welcome everybody. As I said, 3rd of our series of Hub Cures. We have a great speaker and moderator coming up. I want to remind you that the MALSI Plus registration is now open, and it's virtual again this year. We'll be on the Goalcast platform, and it's free. So please go and register for it. Somebody will, put the link in the chat. Also, I want to mention that we are soliciting posters for the poster competition, which, for those of you who've been involved in MALSI, results in some cash prizes. And by the way, last year's poster winner, Concerto Biosciences, went on to raise $1.5 million in a seed round. So getting that exposure can be very valuable. Posters are due by June 15th, so we're looking for predominantly academic spin-outs or soon-to-be spin-outs. So early stage translational science that hasn't quite been discovered yet, by investors, but the platform would allow them to get discovered by investors and other entrepreneurs. So with that, I'm going to hand it over to Joel Bressler, our moderator, for today's fireside chat with Kim Lewis.
Thank you very much, Vineet, and thank you for your support of Northeastern Research. I'll introduce Kim, and then we'll just have an informal chat, and then we'll welcome your questions. Dr. Kim Lewis is University Distinguished Professor at Northeastern, where he heads up the Antimicrobial Discovery Center. The center develops novel antimicrobial therapies to combat both conventional pathogens and biowarfare. As we will discuss today, multi-drug resistant pathogens now kill some 700,000 people worldwide each year, so there's an urgent need for new antimicrobial strategies. Professor Lewis is a finalist for this year's European Inventor Award. Previously, his lab received the NIH Director's Transformative Award, given for exceptionally innovative and unconventional research. He is also a member of the editorial panel for the NIH Director's Pioneer Awards. And while I could keep going, I'll just mention that his 2015 paper in Nature on the newly discovered antibiotic Teixobactin was the most discussed science and engineering publication that year. In addition to his many scientific achievements, Professor Lewis has been involved in founding 5 companies to capitalize on his research. So, Professor Lewis, I invite you to introduce your body of research to our group and describe your current areas of greatest focus. And perhaps we could start with the threat of antibiotic-resistant bacteria.
Thank you, Joel. It's great to be here. It's a big subject, but I will try to compress it. So I think it's useful to reflect upon how we got here into the midst of an antimicrobial resistance crisis we are experiencing at the moment. And keep in mind that in the mid-60s, it was considered that antimicrobial resistance is not a problem, and we've conquered our bacterial pathogens. So how is it that we in 2021 went backwards in time to 1960, maybe, right? And so the way we see it, this is indeed a paradox in our field. In all other fields of biomedicine, the more you know, the better you're able to develop new therapies. There's a positive correlation. And here we don't have this positive correlation. Of course, we know much more than we did in the 60s, and yet it looks like we lost something. So the thing that we lost was a particular source of antibiotics that Selman Waxman introduced to the world, and that is actinomycetes, from which most classes of antibiotics we currently use emerged. You know, the tetracyclines, streptomycins, erythromycins. These are words that I'm sure everybody heard, irrespective of whether you are an expert or not. By the mid-60s, actinomycetes were overmined. It's just like any mine. You know, you mine it properly and it goes empty, and that's what happened. And so since about 1965, we've been looking for a replacement, both in natural products and in synthetics, and it's been a tough road. And there is, I would say, let me speak about obstacles. Maybe Joel then has another question to me regarding solutions. But let me first just tell you why it's been so hard, why it's been harder than in other areas of biomedicine, where there have been enormous advances in developing new therapies, very successful. So first of all, bacteria rapidly evolve resistance, right? Human cells don't do that. So, you know, you develop a therapeutic for asthma, you know, we people are not going to develop resistance to that somehow. So that's sort of one very big distinction. So then that limits the number of targets that you can go after. You can go after targets where resistance is not going to rapidly develop. Another big obstacle is literally the obstacle of penetration. So bacteria evolved what we call multi-drug resistance pumps. The first one was actually discovered in my lab a long time ago, which physically pump out compounds that the bacteria don't like, and those are going to be antibiotics that we're trying to develop. So they pump them out. So you need to, so again, that collapses the universe of molecules that you can develop into drug. And those obstacles have been, you know, a very serious issue. Add to that, overmined actinomycetes, you can go to other groups of bacteria, but bacteria, they like to make stuff that is not useful to us. Oh, I think there's a car beeping, sorry. That's not useful to us. So they love to make, you know, detergents and redox agents and DNA intercalators, and they fight each other with that stuff, right? And that, of course, is completely useless to us. So there's a very big background of that junk through which we have to sieve. So those are the obstacles.
Those are the obstacles. And then, you know, it seems like, as you were describing the paradox, we have increasing knowledge in each of these areas. And yet, you know, we're in a moment where we have hundreds of experimental cancer treatments in clinical trials, right? But only in around 40 or so experimental antibiotics are we having not only a science crisis, but a support for researchers such as yourselves crisis?
Joel, that 40 is misleading. I mean, you're accurate on the number, but that number is made up mostly of analogs of known compounds. So yet another fluoroquinolone, yet another variation on the theme of, you know, tetracycline or erythromycin. And these are like Band-Aids. They give us a short window of respite. So the challenge is to go after truly novel antibiotics for which there is no pre-existing resistance. And for those, the number is not 40. You know, for those, the number in the clinic right now is less than 5.
Mm-hmm.
So, anyways, but you mentioned another thing. You mentioned that another challenge is indeed, and not a science. So I spoke about scientific challenges. Of course, we all problems have solutions, right? So of course we'll work on it. We'll figure it out, yeah, as we do, as we go ahead. But there is another challenge, which has nothing to do with science, or almost nothing to do with science. And that is the challenge of the market, the reality. So doctors, correctly fearing the rise of resistance, came up with what they call good stewardship practice. Good stewardship practice means that if you get an infection, I don't know, staph MRSA infection, nasty thing, doctors will typically put you on vancomycin. And if there's resistance to this compound, well, then they'll put you on daptomycin. And if there's resistance to that, they'll put you on linezolid. And by that time, considerable number of patients are dead. So there's a distinct downside for the individual patient from good stewardship. The good stewardship was introduced in the interest of the population, not of individual patients, right? So that's a peculiar ethical issue here, because the doctors give an oath to patients, not to the population. Something to keep in mind. But because of that, what it means is that if you are discovered a new terrific compound and it got approved, it means that the doctors are going to put it on the back burner. Put it on the back burner means that there will be very little in sales, and it will be exceedingly difficult to recoup the billions of dollars that go into development. And so that is the reason why several antibiotic companies just went bankrupt. With not entirely novel compounds, so we still don't have a test case for that, but for analogs of existing compounds, 2 companies went bankrupt recently, and that put a chill on the investment, infrastructure in terms of supporting antibiotics. Investment largely dried up, but there's hope. So England, for example, introduced a new model of how hospitals pay for medicines. If you have a new, truly novel compound, they will pay for it, for the ability to use it, irrespective of the volume of sales. So it's a subscription model, right? They will subscribe and pay up front. I think it's a terrific idea. Here in the U.S., Congress is contemplating a persister legislation where there'll be a very sizable upfront, billions of dollars upfront payment for a novel compound. And I read with interest the wording of that. There's one property that the compound has to have, apart from the obvious. It has to be interesting. I wonder what our congressmen and women think is an interesting compound.
Well, you as we were in the green room, you teased me a little bit, because I asked you if now, in addition to succeed in the antibiotic space, you not only need to be a scientist and a business person, but a policy wonk as well. And you teased me by saying you had some thoughts about that aspect of the business.
Actually, the main thought I had, I didn't share with you guys. The main thought is the scientific solution of the marketing problem. And here it is. So imagine that you have an antibiotic to which there is no resistance, and there's not going to be resistance anytime soon. If you can demonstrate that experimentally, then the good stewardship practice no longer makes any sense. Actually, it becomes unethical, right? Not to put forward as a first line of defense the new antibiotic to which there's not going to be resistance. And I see, I don't know, Joel, if we are segueing into, Novobiology, et cetera, because that brings me to one of the compounds we discovered that has such properties.
Sure, yes, please. That was your first company, is that correct? Novobiology?
Yeah, that's a company we started with Slava Epstein, my colleague at Northeastern. And the idea behind that company was very simple. If actinomycetes are overmined, let's find a source that has been truly virgin, untapped in terms of microorganisms. And these are uncultured bacteria. So happens that about 99% of bacteria on this planet don't like to grow on our petri dishes. So we call them uncultured or unculturable. And so we developed a method to grow them, actually. And it's just extremely simple. We grow them where we know that they will grow, which is their natural environment. So essentially, you take scoop up some earth, You dilute it with, with agar, and you sandwich it between two semi-permeable membranes. And that contraption, we call it diffusion chamber, goes back into the earth environment. Now everything diffuses through that chamber, and the cells that are sitting inside, they don't know that they've been tricked. And so they produce colonies. And so we can grow in pure culture. Now, bacteria in their natural environment, isolate them, adapt them to growth in the lab, which we call domestication, and use them for antibiotic discovery. So from that approach, the company Novobiology, in collaboration with us, discovered Teixobactin. Well, they discovered a bunch of compounds, but this is the lead now. And the amazing property of Teixobactin is that there is no resistance to this compound. And because it hits targets that are not mutable, two different targets, neither of which is mutable. They're not proteins. They don't mutate. You cannot mutate them. And in the space of the last 6 years, there has been any reports of resistance. This compound is now at late stage preclinical. We're supposed to go into phase 1 by end of this year, but because of COVID, there's a delay. So hopefully it will go into phase 1 next year. There's a limitation to this compound. It only hits gram-positive bacteria, not gram-negative. But still, very nasty bugs like Staph MRSA, that's where the primary focus is. So here's an example of such a compound to which there is no resistance. But you cannot really count on that. That's a very lucky hit. But what you can, I think, count on in terms of a general solution to the AMR crisis and to the marketing problem is combining two novel compounds. Let's say you're doing well, you have more than one novel antibiotic, and there's resistance to them. These are not like Teixobactin. These are more conventional compounds, hit protein targets, there's resistance development. But now you have two. If you combine two, then there's not going to be resistance anytime soon. So the math is very straightforward. It's like flipping a coin. The probability that there'll be resistance to one antibiotic is typically 1 in 10 million, so 10 to the minus 7. If you have two such compounds, it's 10 to the minus 7 times 10 to the minus 7, 10 to the minus 14. So now we've got astronomical numbers. So that, I think, will provide a general solution to the solving the AMR crisis, pulling us out of the rat race against our pathogens, and also will provide the solution to the marketing problem, because now your combination should be the front line of defense rather than kept in reserve, precisely because resistance is not a concern.
Fantastic. Let's stay on Novobiology for a moment. If there are others in the audience that are looking to find, to test, to market new antibiotics, can you crystallize some of your experience at Novobiology and Teixobactin and tell them some of what they're in, what sort of roller coaster ride they may be in for?
Right. So, yeah. So it took us about 10 years with Novobiology to get to Teixobactin. And though we're doing better in terms of discovering novel molecules, we still have an enormous problem that people in the 70s, 80s, and 90s who pulled out of antibiotic discovery had with actinomycetes. This enormous background of compounds that are not useful that I mentioned. And so because of that problem, we haven't seen much compounds that hit gram-negative bacteria. That they have more powerful pumps that pump stuff out. So these are creatures like E. coli, common E. coli, but this common E. coli acquired multi-drug resistance, or Klebsiella pneumonia, that now the death rate, if you have an infection with multi-drug resistance Klebsiella pneumonia, mortality is 40%. It's like pre-industrial times. So the question for us was, what do you do about it? And how do you yourself discover or share some useful knowledge about how to go about this, as Joel was posing this very useful question. So I'll tell you, it's a simple problem, really. It's a simple problem with a relatively simple solution. So here's a problem. You have your drug discovery process. So you take a gram of soil, you isolate bacteria by whatever method, culturable, unculturable. You got your bacteria. And then you test them for ability to make compounds. And then most of the compounds are going to be junk, and that's what you spend lots of time on. And so the profession, and my lab also, spent a lot of time trying to figure out that downstream process and how to resolve those bottlenecks. But then I realized that I'm probably thinking about the wrong problem. Because you see, the main bottleneck is at step number 1 that nobody likes to talk about because it's a seemingly unsolvable problem, so it's the point. And that is when you take your gram of soil with bacteria, you assume that there's a wonderful producer of a novel antibiotic there. And of course, you don't know that. And so it's entirely possible that you spent your life chasing these grams of soil, and you never had a producer of new compound in those grams of soil. And you don't know that, which is a maddening, consideration. So there's a straightforward theoretical solution to that problem. Don't screen grams of soil, screen the planet, the entire planet. Of course, there is a bit of a practical issue here, how you're going to do that. Because it's a theoretical solution, yes? So how do you do that? And it seems like a rather obviously, absurd proposition from an engineering standpoint. But there is a solution to that problem. And I think this is how it works. I think that the planet Earth has already been screened and multiple times by bacteria that make antibiotics. And the way they do that by horizontal transmission of DNA. So DNA floats around the globe. Everybody leaches it out, including us, and all bacteria and all fungi. They leach DNA. DNA is floating around, a cloud of DNA, if you will. And everybody can pick it up. It's called horizontal transmission. So all organisms, including people, pick up pieces of DNA and insert it in their genome. A typical group of bacteria has been around for 100 million years. So now they have their certain preferences for certain antibiotics, and they have 100 million years to screen DNA floating around planet Earth for stuff that they want. So then the challenge for us people is to find a group of bacteria that has the same requirements for antibiotics as we do, right? Due to their ecology, to the whatever. It's the same short list. It's sort of common sense, obvious list. The compound should be non-toxic, potent, hit important gram-negative pathogens. That's our number one concern at the moment. Have good in vivo efficacy against pathogens, so we work in an animal. So that led us to an interesting group of bacteria that live as symbionts in the gut of nematodes. And these are entomopathogenic nematodes. So they attack insects. So you can, by the way, you can buy them on Amazon, which we do. So you buy these nematodes on Amazon, and the reason Amazon sells them is because you can put them in your flowering pot, and that will protect your favorite geranium from insect infestation. Right? So the nematodes carry Photorhabdus bacteria. Once the nematode gets into a caterpillar, releases neurotoxins, so paralyzes its future food, releases Photorhabdus bacteria. And the bacteria start making antimicrobials. And of course, these antimicrobials have to be non-toxic to the nematode, which is an animal. They have to be able to travel through the tissues and kill, freeloaders that want to attack that corpse. And the main competitors in that environment are gram-negative bacteria. So that's our list. And they originated 375 million years ago. So that's how long they had to act together, if you will, and to assemble a set of antibiotics for their purpose. So now we're mining these bacteria, and one of the things that we discovered soon after we started looking at that group was darobactin, a novel class of antibiotics. It satisfies exactly that list. It hits gram-negative bacteria, is non-toxic, efficacious in vivo, all the good stuff. Hits E. coli and Klebsiella that I was mentioning to you. And so that is an example of doing something that is productive, sort of hitchhiking on the screen of planet Earth, that has already been done before us.
Looking in novel places to find novel compounds, essentially.
Yeah.
Because the most recent gram-negative antibiotic before that was a full 60, it was in the 60s, right? It was a full 60 years ago.
Yes, fluoroquinolones. Yes, the last class of antibiotics introduced into medical practice, fluoroquinolones. So you probably are familiar with ciprofloxacin, right? That's sort of a common representative of that class. That was in the 60s.
Fantastic. You've touched on Novobiology and Daros. You have a microbiome spin out from Northeastern as well called Holobiom. Maybe if you'd like touch on that for a moment or so.
I'll mention that briefly. So one thing that we're trying to do is understand why uncultured bacteria don't grow on our petri dishes, including uncultured bacteria in our microbiome, in our gut. So important symbionts. And so we found a number of growth factors that help them grow in our petri dishes. And one of those growth factors was completely unexpected. It was GABA, gamma-aminobutyric acid, our important neurotransmitter. And there are bacteria that release it and bacteria that consume it, right? So there's a whole story going on around GABA. And so my postdoc at the time, Phil Strand, decided that he'll start a company based on formulating bacteria that make GABA and put it in a pill for people who have trouble sleeping or have visceral pain. And there's now Holobiom has a collaboration with Johnson & Johnson to develop that. So that's sometimes science takes you on completely unexpected detours.
Absolutely. I mean, talking about unexpected detours, a lot of you've had research in persister cells as well, and I know that's taken you into a number of different regions, such as Lyme disease and other areas. And I believe you're commercializing that as well now, an aspect of it in FlightPath.
Right. So I'll briefly mention about Lyme disease. I don't know how well we're doing with time.
We're doing fine.
Okay. So Lyme disease is a huge problem, and I was very intrigued. Not only wanted, obviously, to do something to help patients with Lyme disease, but also I was intrigued with this very difficult disease. And one of the things that we were able to do in my lab is identify a compound that very selectively and potently kills Borrelia burgdorferi that causes Lyme, because we had reason to believe that dysbiosis in the microbiome may be contributing to or causal to more chronic forms of Lyme disease. And we recently published a paper showing that the microbiome is different in people with post-treatment Lyme disease syndrome. So if that's the case, of course you want antibiotics. It's not going to be a broad spectrum to additionally screw up your microbiome. To treat Lyme, you want something selective. So we found such selective compound. It's very efficacious in animal models. It's orally available. Highly selective, doesn't harm the microbiome. So that is something that we licensed the FlightPath and hope to develop into a dedicated drug to treat Lyme disease. Hopefully without the unpleasant consequences of developing a chronic condition.
Absolutely. Fantastic. Perhaps I'd like to ask you a question that's sort of 90 degrees to everything that we've talked about up to now. Both you and your collaborator on the iChip, Slava Epstein, were trained in the Soviet Union, were trained in Russia. And I was intrigued in your nomination for the European Patent Office Invention of the Year. It talked about how your Russian training might have fostered a different perspective in microbiology. What is that different perspective, and how do you think we might foster some more of that sort of iconoclastic thinking in the U.S.?
Right. That's a good question. One thing I definitely do not recommend, and that is, it was useful for me and for Slava, I don't recommend it, and that was lack of resources. Okay?
So it wasn't scarcity, it was more...
Right. You're extremely limited in reagents, in instruments. And so you need to spend a lot of time thinking about what is it exactly that you want to do. And so that, I don't recommend it. But that also means that there's an enormous emphasis on the intellectual side of things, right? That was sort of in the culture that we essentially were trained in. But there is another thing that I do recommend and that I use all the time. So Russian engineers, especially the guy, the last name Altshuler. So Altshuler was an inventor who decided to formalize invention. So he thought that he invented how to invent. And he published a thin little book on that, How to Invent, which I read when I was an undergraduate student. It made an enormous impression on me, and I use that all the time. So for example, what I told you about screening planet Earth, that comes directly out of Altshuler. So what he was trying to do, he realized that our mind doesn't do two different things simultaneously very well. We do not think creatively and practically at the same time very well. So our common sense always tells us where not to go because it's not going to work. So don't think in that direction. So he decided to separate those two things. And he said, why don't you do this? Formulate a perfect result that you want. Don't worry about practical reality at all. So essentially turn off your common sense. And then see, once you formulated that, then you can start asking the question, well, how to do that practically. And he said, and if you formulate something and it sounds absurd, you are onto something. Right? That was his message. So I'll give you one example the example comes from, he has examples in his books. Oh, by the way, the method is internationally fairly well known in engineering circles. You can read it up, check it out on Wikipedia. Biologists and basic scientists don't use it. I recommend you to check it out because I use it all the time. Most of my inventions and papers come out right from that recipe of the perfect result. So it's called TRIZ, T-R-I-Z. It's an acronym for some unpronounceable Russian words, right? Don't worry about it, but Wikipedia has it. Not a bad write-up. Wikipedia, unfortunately, the write-up is boring because it doesn't give you examples. I'll give you one example.
Please.
So the example, engineering, right? That's not biology. So the example comes from a guy who I happen to know, Simon Litvin. So Simon Litvin is, he lives here near Boston. And he's a CEO of a company based on TRIZ. It's a multinational company which has as its clients, Intel and General Electric, General Motors. It's serious operation, and they invent for hire. You can hire them to invent. That's what they do. So the company is called Gen TRIZ. And here's an example of their solution of a problem using this formalized invention process.
Yes, please. Sure.
So here's so there's a company in Japan, it needs to build an automated assembly line, and that assembly line will be making some gadgets out of steel, and they have to be machined. And the problem is that when robots machine them, there are pieces of metal flying around, and those metals, drop down and jam up the machinery. Question: how do you solve that problem? There want people there. So, well, the ideal result is to have those things, instead of dropping down, going in the opposite direction. That's your ideal result. So then you start thinking, what's the practical solution? You can suck it up with negative pressure. You can put a magnet. That's very expensive. So this is a solution that they came up with. Take your machinery and mount it on the ceiling. And then everything is going to fall down on the floor. And since there are no people, it really makes absolutely no difference where your machinery is, right?
Problem solved?
Yep.
Fantastic. Well, I'm going to invite the audience at this point to ask questions and invite Rachel. I think I saw at least one question that had been upvoted. If you could present that to me again. Here we are from a Michael Hallstrom. Michael's question is, how could microbes treat microbes? For example, could a monoclonal microbe be isolated, scaled up, and used to treat Lyme disease? The downside of antibiotics is the decrease of beneficial gut bacteria. Is that something that you and your colleagues have looked at?
Okay, that's an excellent idea, actually, which has its practical manifestation in Seres Therapeutics and several other microbiome companies. So the most advanced microbiome product at the moment, which is now just completed phase 3 trial out of Seres Therapeutics, is a concoction of Clostridia bacteria in the form of spores that is given to people who suffer from Clostridia difficile infection. Now, the spores are symbiotic Clostridia that will outcompete the pathogenic Clostridia difficile. That's the idea, right? And it works much better theoretically, and I hope in practice, than antibiotics, because what happens with antibiotic treatment, you give antibiotics, Clostridia forms spores. Spores are not killed by antibiotics. So when spores survive, you wipe out the whole microbiome. The only thing that is left in your gut is spores of Clostridia difficile. All right, not a good result. So your antibiotic goes away and you just relapse. So the Seres Therapeutics approach restores your microbiome while killing Clostridia difficile. It's a brilliant solution to the problem. Now, as to whether you can do tricks like that for systemic infections, that I'm not sure. Because we have an immune system, you start introducing microbes, our immune system will be unhappy.
It's great to have that series example, though. Rachel, other questions from our audience? From Holly Suter. I agree completely that combos should be standard of care. What needs to change in the regulatory process and the market to make that happen?
The regulatory, I would say, so right now it is possible to introduce combinations of antibiotics. So, for example, ampicillin and clavulanic acids, so the inhibitor of beta-lactamase is a combination. So combinations are periodically, introduced. Are periodically introduced, but industry doesn't like to do that because the regulatory process is fairly arduous. It's more expensive. It's harder to do. We don't have too many novel compounds, so it's more like a wish. But I think one thing would be very useful to do if FDA got its marching orders that this is what you need to support. This is where you need to ease the path and think creatively how you're going to do that, rather than just to say, okay, give me your proposal and we'll poo-poo it or whatever,
So some emphasis, if you will, at the highest reaches of government or the highest reaches of public health in order to make that happen.
Yeah.
Excellent. Rachel, others? From John McLennan. Could you elaborate on how you, quote unquote, fool soil bacteria to replicate and then to domesticate them?
Yeah, I know how to replicate. I understand. That's simple. You put them in essentially what is a dialysis bag. So you put them in a compartment enclosed with a semi-permeable membrane. So cells cannot move back and forth, but small molecules can through small pores of that membrane. And then you stick it back in soil, and everything diffuses through that contraption. So the bacteria don't know that something happened to them, right? They receive absolutely the same stuff that they would when they were there in soil. They don't know that their environment is covered with membranes. So of course they grow and form colonies. So that's straightforward and easy to understand. Domestication, we do not understand. What we did discover is that once a colony grows inside a diffusion chamber, with a high probability it will grow on a petri dish in the lab on regular media. So it looks like formation of that population, the first colony. That's where the barrier and the bottleneck is that we resolve. We have some ideas how domestication works, but I don't like to bloviate about ideas that are not supported with evidence.
Thank you so much. Our time goes short. Rachel, is there one other question that we could pose to Professor Lewis? And if so, present it. Very good. From Carmela Mascio, I hope I pronounced that correctly. Thanks from me and from Carmela for speaking about such an important topic and expresses the opinion that we need new antibiotics to combat these resistant infections. She would like to hear your thoughts on also prioritizing innovation in infection prevention in addition to antibiotic discovery.
Yeah. So sure, I agree with that. That's not an area that I understand professionally. Of course, I know something about it. So there's abuse and misuse of antibiotics in animal husbandry. Also in some developing countries where you can get antibiotics over the counter very easily. There's overprescription of antibiotics. And if we were able to solve those problems, our life would have been easier. Some problems we can solve. So already in the developed countries, there's virtually little or no use of antibiotics as growth promoters, for example, in animal husbandry. There's this good antibiotic stewardship that I mentioned. But unless there are serious political, changes in developing countries, I really don't see how that will change things. So I think the focus on discovering and introducing novel effective compounds is probably the more practical way forward.
Thank you. And as we emerge from a pandemic, an unforeseen one, but some people would tell you a predictable one, we face something of the same dynamic here with the shortage of antibiotics. And, we thank you so much for your work. It's so important that we have these novel compounds, both for gram-negative and gram-positive. And I, for one, have really enjoyed being able to speak with you about it today. And thank you so much.
Thank you, Joel. Thank you, everybody, for listening.
Thank you. Now, I don't know if Priya or Vineet will come on and usher us into the next stage of the presentation, but I would invite that. There's Priya. Thank you so much for hosting us. We're really very grateful to you.
Oh, thank you so much, Dr. Lewis and Joel. That was a fascinating conversation. We enjoyed it tremendously and learned a lot from all of you, both of you. And just a few reminders before we head back to networking. Next up on the June 8th Hub Cure series, we have Jeffrey Karp, a professor of medicine at Harvard Medical School, who will be joining us for It's Not Failure, It's Intentional Iteration. So if you're intrigued, do join us. And Jenny Rowe will be moderating that conversation. And secondly, if you joined us late today, registration for MALSI, which is on July 22nd, is now open. We have an exciting keynote speaker and an amazing lineup of panelists. We're also accepting poster applications at this time, so please don't forget to submit yours. So with that, please enjoy networking. Thank you again, Dr. Lewis, and we hope to see you in the networking area. Thank you.
Thank you.
Thank you.